My name is Lucie and I work at ITQB Nova in Portugal. And here I will show you the energetics of palmitate complexed with BLG using constant pH molecular dynamics. Beta-lactoglobulin, or BLG, is a milk protein and is the major component of bovine milk. It is known also to cause an allergic reaction in some individuals. It also has an hydrophobic cavity where small hydrophobic ligands can bind, and although its function remains unclear, due to this property, it is believed that it may act as a specific transporter. It can also form mainly a monomer or a dimer in a pH-dependent way, and this association is by electrostatic interactions. This monomer-dimer equilibrium is affected by several factors, such as the protein concentration, the ionic strength, and being one of them, the pH. And at around pH 5.3, it's isoionic point, and it is known that the dimerization is favored near this isoionic point. In this hydrophobic cavity, there is a loop called AF loop that it can be in a closed or in an open position according to its pH. That is, at pH lower than 7.5, the loop is considered to be closed and ligands cannot bind, but when the pH increases, the loop opens and ligands can bind. And this gating mechanism is affected by the protonation of glutamate 89, and this transition is called the Stanford transition. The ligand binding is strength depends on how long it is aliphatic chain, being that approximately longer ligands have a stronger binding. And actually, I'm saying approximately because just before coming here, actually, I found a paper that also suggests this, but when looking into it, their binding free energies, their differences between different ligands were very minimal and within its experimental error. So still need to look more into detail. Either way, this ligand is mediated by van der Waals interactions in the hydrophobic tail and by electrostatic interactions with its carboxylic head and with the nearby residues. Here I focus only on the palmitate using constant pH molecular dynamic, and since this was already, the methodology was already explained in the previous days, I think that I don't need to do it again. But basically, I use the stochastic method, the same used by António and Miguel. And very briefly, we can sample the protein conformation using MD. From time to time, this simulation is stopped, and then a Poisson-Boltzmann Monte Carlo calculus is done to sample the protonation states. I will focus mainly on the simulations done with the palmitate complexed with the BLG, the holoform, but I will compare it with the previously done simulations using BLG alone, the apoform. They were both done in very similar conditions. In both, I have used the monomer and the dimer in a pH range from 3 to 8. For the apoform, I have done a total simulation time of 100 nanoseconds. And for the holoform, I have a little more sampling, 200 nanoseconds. In the apoform, we have used the closed and open crystallographic structures, the one that have the loop in the closed and in the open position. And in the holoform, I only have used the open structure because palmitate is bound to BLG. Passing to the results, we have here the protonation of the monomer and the dimer for both the apo and holoforms. We also have the titration curve of an experimental study, and we can see that Our predicted charge is in agreement with the experimental one, with minor differences at higher pHs, and also our predicted isoelectric point 5.1 is also in agreement with the experimental one, 5.2, 5.4, depending on the study. And for the holoform, we have isoelectric point of 5. So by using only the titration curves, we can obtain the variation of a free energy along a pH range by taking advantage of a thermodynamic leakage relation. Here, I will first show you the dimerization free energy using only the difference of the dimer and monomer protonation curves. And this is a quite interesting approach, since we don't need to study the association process itself. We can study the monomer and dimer independently. Basically, this is done by, with the difference, we have an area that is computed in an integral. Since this is a relative free energy, we use as our pH of reference the starting pH, pH 3, and we need to solve this integral. This could be done using simpler common methods, such as the trapeze or the rectangle method, but because we have very few points and they are all spaced apart and we don't have intermediate points between them, this will not give us a very precise result. So one solution is to instead do the sum of the fit of Anderson, Hasselbalch, or Hill curves to our points, as it was already done by others. But if the points deviate a little, as it's our case, this will not be as accurate. So our solution was instead to use the slopes from the titration curves using the protonation fluctuations by using this thermodynamic relation. So basically, for each curve segment, we can obtain two slopes and we have 2 points, and this will have 4 parameters that can be computed in a third order Hermite interpolation spline, and with this we'll have an integral that becomes a fourth order polynomial. We have here the dimerization free energy for the apo and holoforms. So our method is depicted with the line in black. We call it the spline method, with the corresponding arrows in gray. And to compare it, we also did the fit to the Hill curves here in green. We also have some points from different experimental studies in which they all have the same ionic strength and temperature than are simulations. We have some dispersion between them, but this is probably due to the different experimental techniques used. So since this is a relative free energy, we only know the shape of the curve. We don't know the position. This is why we have fitted our curve. We did a vertical fitting to the experimental points. And we can see that the dimerization is favored around the isoelectric point, as it was previously experimentally in the literature observed, and that our predicted trend is also in agreement with the experimental ones, mostly at higher pHs. At low pH, the prediction is not that good. And for the holoform, I still wasn't able to see in the literature if we have experimental data. Until now, I still wasn't able to find anything. This is still a preliminary ongoing work, but from the shape, we can see that the dimerization is favored around also the isoelectric point. And from what I've seen in the literature, it seems that the binding also favors the dimerization. We also look into the contributions that each individual residue has to the dimerization free energy, because since the the protonation curve of the total protein is the sum of the protonation curves of the individual residues, then the global dimerization free energy is also dependent on the dimerization free energy of each individual residues. And by looking into these graphs, most residues contribute very little to the pH dependency. However, there are some residues that favor association and or dissociation depending on the pH. I have selected some of them, and we can see, for example, that in the interface, we have some residues that contribute as it is expected in the dimers interface. For example, this aspartic and this histidine. And we also have some residues that also contribute but are more far away from the interface, as it is the case of this glutamate 89, the one that, as I've told you before, is associated with this gating mechanism called the TAN4 transition. By using the same rationale, we also were able to obtain the ligand binding free energy. In this case, we have used the protonation differences of, we have done the difference of the holoprotonation with the sum of the apoprotonation and with the protonation of palmitate in solution. And by looking into this thermodynamic cycle, we basically have two monomers that gives a dimer, and with this we have obtained the dimerization free energy of this apoform. By adding a ligand, we were also able to obtain the dimerization free energy of the holoform, as I've just show you. And since we have these two types of different simulations, we are then able to obtain the ligand binding free energy. It's presented here, and basically I have already found some experimental data in the literature, but this requires a lot of time and patience to see if their experimental conditions are similar to our simulated ones. And I still wasn't able to do this, but from what I've seen, this ligand binding free energy is favorable, is around minus 8 to 10 kilocalories per mole, which makes sense since the palmitate is hydrophobic. And looking, we can see that in both the ligand binding free energy decreases until more or less 6, 7, with a bigger increase in the monomer than in the dimer. But I find this quite interesting, since it's also here where the TAN4 transition occurs and where the palmitate can bind. And compare it with the yield curves, their shape is not it's similar to ours, and the trend also it is. If we look to the contributions that each residue has for both the monomer in the dimer, we can focus first on the monomer in some selected residues. They are depicted here, and in red there's the loop associated to the TAN4 transition, and in green the palmitate. And for example, this aspartate here, it contributes, but it's far from this hydrophobic cavity. However, these ones are located near it, and again, the glutamate 89, it contributes to this binding free energy. Also, palmitate as a big contribution. It's more favorable until pH 5, which makes sense since it titrates at this pH. In the dimer, besides the residues that both contribute in the monomer and dimer, as depicted here in blue, it is interesting to see that there are some residues, like mostly these two ones that are present in the dimer and not in the monomers, and they are located in the dimer's interface. So this seems to be in agreement with the fact that the binding favors the dimer's interface. So this is still an ongoing work, but in summary, we have the absolute values of the dimerization free energy of the apoform. But for the other three energies, we only have relative values. From what I've seen, I believe that we will be able to obtain experimental data on the literature for these two ligand binding free energies, but I am not quite sure about this one. So, but basically, since we have three independent profiles, we can obtain the fourth one. To conclude, so dimerization is favored around the isoionic point, as was experimentally observed in the apoform. The ligand binding is favored around the 10-4 transition. And although we can conclude about the dimerization and ligand binding affecting each other, from what we've seen in the dimers residues that contribute the most, they seem to be related. Finally, I would like to thank you all for your attention. The Molecular Simulation Lab, António, that is here, and Sarah, and also the funding. Thank you.
Thank you. Any question?
Could you go back to the one with the red dots on it, your experiment versus the calculation? So what I'm curious
This
about
one?
is whether if you plotted the error, the difference between your dot and the line versus pH, if you'd have something that had a slope, some very clear correlation, and if that would give you some idea of maybe what's going on, because you're above the line at high pH and below the line at low pH. So it seems like
in this case here.
Yeah, but also above the line at high pH. So there's something kind of systematic that maybe you're missing that maybe you could get a clue from if you think about. But it's very lovely, and congratulations.
Yes, maybe also they have a lot of dispersion between them. So yeah, but we can't. I think that the prediction here is better, but at low pH we can't really understand why.